So the first of the four proposal types is the so-called large-scale EMSL research program. This funds proposals, user proposals for up to 2 years. It advances scientific understanding in each of EMSL's 3 science areas, which I've highlighted here below. In green, blue, and orange, environmental transformations interactions, that's what we're talking about today. And then functional and systems biology. So there will be, as Jenna has said, there will be a future webinar for that, and also computing analytics and modeling, and that'll be covered in a similar future webinar. So we're going to be talking about environmental transformations interactions today. There will be 4 separate call topics that'll come out with a large-scale research call. They will, by and large, align with one of the 3 integrated research platforms that we have. And in today's talk, you'll hear Swaroop China and Amir Akami introduce 2 of those 3, which actually are new. So this is a really cool opportunity to kind of get in at the beginning. So we also tend to highlight novel applications of EMSL capabilities. So the proposal call opens on December 14. Letters of intent are due on February 9. And yes, if you're wondering, a letter of intent is required in order to submit a full proposal. The deadline is March 23rd. I just want to emphasize 2 other things. First of all, the vast majority of these letter of intents are actually approved. So this is actually, and the second thing is that's because we have a very interactive proposal review and submission process. We use the LOI to understand what you want to do and provide really valuable guidance back to you before you go to the effort of writing a full proposal. And it is a very interactive process. Please, if you decide to write a proposal, feel free to reach out to our contacts to do that. That's the way we like to do business. The second type of proposal call supports collaborations between EMSL and JGI, has a rather fancy name of FICUS, which stands for Facilities Integrating Collaborations for User Science. I can never remember that, so you don't need to. But the cool thing about this for you is that you write 1 proposal and you get access to 2 facilities. Again, they're funded for 2 years. You are connected as the user, you're connected to capabilities at EMSL and at the Joint Genome Institute. And in addition to those, you also have additional capabilities available through our partners, including the National Ecological Observatory Network, which you may know as NEON, the Biosands Beamlight at HIFER through the Center for Structural Molecular Biology, and also the eBerLite program through the Advanced Photon Source. So the focus areas will come out when the full call opens on January 9 or thereabouts. Letters of intent will be due in mid-March, and full proposal will be due on May 4. All right, so the third of the 4 proposal types is another collaboration, in this case, another FICUS specifically. And in this case, it's to have joint access to capabilities at EMSL and at the Atmospheric Radiation Measurement Laboratory. So again, funded for 2 years, connects researchers with molecular characterization capabilities here at EMSL and sample collection capabilities at one of the ARM mobile sites deployed around the US, as well as science capabilities. Again, the specific focus area will be announced when the proposal call opens in just a few weeks on the 14th. Letters of intent are required if you want to submit a full proposal, due on February 9, full proposal due on March 23rd. And then the 4th type of call is actually not for a full proposal. It's actually the first of a brand new type of solicitation we're going to put out. It's called MONET Soil Sample Call. MONET stands for Molecular Observation Network. And in this case, there's 2 major things to understand. We're asking for the community to participate in this community science project, to submit core samples and grab samples using a standardized procedure that we'll send you. And in return, you get a tremendous amount of information. We actually have a standardized workflow that produces 24 different data types, notably including high-resolution organic matter composition, the pore network structure, metagenomes, hydraulic properties, and a big long list of other biogeochemical parameters. This is free. So there's so many data types here that graduate students, postdocs, faculty who are running research programs that are at all related to soil ecology can get a big boost by just submitting a sample. We do it all. We do the analyses. We're trying to build up a database. It's a cutting edge new database. The data will be publicly available upon deposition. And so you can send a sample, you get a ton of information, and it helps build up a novel, a molecular soil property database that will support new climate modeling and land atmosphere interaction modeling. So we'll initially release 2 topic areas. One will be soil function, and the other one will be rapid response, so that we can respond to science opportunities created by a short-term transient phenomena like flooding. We're hoping it will open on February 1st. This is a new type of solicitation. It's a little different from the other proposals. And in fact, exactly a month ago, we had a webinar on the Molecular Observation Network. And I encourage you to visit our webinar library, where you'll see lots of other cool webinars there, but including the Monet one. So if you're interested, please go look at this. For more information, visit the EMSL website, and you can also contact us at emsl@pnnl.gov. And with that, I'll wrap up.
Hi there, sorry. I was just trying to find my Zoom. Here it is. Okay. So let me go ahead and share my screen. Oh no, I can't share the screen, so I'm just going to read off the slides. Okay, so my name is Jeremy Zucker, and I'm going to be talking to you about a particular algorithm called metabolic control analysis. And although the title says for fungi, this can be used for any kind of system in which you are trying to develop a microbial cell factory for improving titers, rates, and yields of a particular bioproduct. Next slide, please. Well, okay, there we go. So, in particular, this algorithm can help you identify which native or potentially non-native enzymes have the largest impact on bioproduct titers, rates, and yields. And what it requires is simply knowledge of the genome scale metabolic network, and it requires multi-omics measurements, in particular metabolomics, proteomics. If you happen to have fluxes, it'll use that too. And there are also extensions to this algorithm that can even use post-translational modifications such as phosphoproteomics. And what it returns as output is basically a ranked list of enzymes, which, if you were to perturb them, would increase the titer rate and yield. So some enzymes, it'll say if you increase the expression, it'll improve titers. Others, it will say if you decrease this enzyme, it will increase the titers or rates or yields. And it'll also quantify the uncertainty associated with that particular prediction. Next slide. Okay, so like I said, the inputs for this are multi-omics measurements for different conditions relative to a reference strain. So the idea is that you would have multiple strains available, and there's one of those strains that grows the fastest or that produces the product the most, and you want to use all the other slower growing strains, and you're basically going to compare the multi-omics measurements of each of those strains against the fastest growing strain. And that will give us the clues that we need in order to be able to answer the question. So the assumptions that this algorithm uses, it assumes that that the organism is in some kind of metabolic steady state, and that's needed in order to constrain these reaction fluxes. Another assumption that it makes is it uses ln-log kinetics to predict the reaction fluxes from metabolite enzyme measurements. That the reason why it uses the ln-log kinetics is it turns out to be possible to solve for steady state in a single linear solve. So for performance reasons, it makes sense. But I'm going to show you some evidence that it's not a bad assumption to make when you are near steady state. It also uses knowledge of reaction stoichiometry to generate prior beliefs about the effect of change of each how much each metabolite affects each enzyme. And so that is known as an elasticity, and I'll talk about that in a bit more. But I also want to give you some caveats. The input perturbations are restricted to different nutrient media. So you can swap out a different nutrient media and compare different strains that way. And the different strains can't be completely different gene knockouts or introductions of new genes. They basically are all, they must be like overexpressed versions of the same strain. So maybe some genes are overexpressed or some are repressed, but they can't be knockouts or a brand new addition of a gene. Okay, next slide, please. So just to get a little bit more detail, so the idea here is that if I have a reaction network that's shown on the left side here, then what we want, then what I mean by an elasticity is that there's this metabolite, and we want to know is if you increase if somehow this metabolite were to be increased, then would that have an effect of increasing this reaction flux or decreasing the reaction flux? If it has no effect at all, then we just assume that the effect of that metabolite on, say, this reaction is 0. But in this case, we see that the greater this metabolite, the greater the concentration of this metabolite, the slower this reaction will go. Whereas in this case, the larger the expression, the larger the concentration, the faster this reaction goes. In this case, if there's a non-zero link here, it means that this must have some kind of allosteric effect on reaction C. And so we can take all the reactions, we can take all the metabolites and create an elasticity matrix, which is hopefully fairly sparse because most metabolites only participate in a few reactions. And we expect that the likelihood that a particular metabolite is going to have an allosteric effect on a random reaction is pretty small. And so the way we represent that uncertainty is we say, well, we don't know exactly how much increasing the metabolism, increasing the concentration of metabolite affects a reaction when it happens to be the product of a reaction, but we know that it's probably negative in the sense that the more product you have, the slower that reaction is going to go. Whereas if you increase the reaction, the concentration of a reactant, then we anticipate that it will have a positive effect. We just don't know how positive. And likewise, the likelihood that a metabolite affects some reaction that it's not a participant in is fairly low. And so we assume a very tight distribution around 0, but we allow for the possibility that the data could make us think that if the data supports it, then we allow for the possibility that a metabolite can allosterically affect an enzyme. Next slide, please. So just to give a very simple example, we take a simple reaction where A goes to B plus C. Then the way that we can represent that, the reaction rate that we are modeling is that the flux, that's V, is a function of E, which is the enzyme concentration, plus the concentrations of each of the metabolites. And then A, B, and C represent some unknown parameter that we're trying to solve for, that we have some uncertainty about, and we want to generate a posterior distribution based on additional data. And so what we're showing here is that this elasticity, these A's, B's, and C's, have a direct interpretation as the slope of this ln log line. And as you can see, there exists a Michaelis-Menten curve that is fairly similar to the ln log curve. The Michaelis-Menten is most likely the kinetic law that is governing each of these reactions. But you can see that at least near steady state, that the ln log approximation and the Michaelis-Menten curve are actually quite similar to each other. As we get further away from, as we get, say, closer to V max, then these will start to diverge because ln log can continue on for infinity, whereas Michaelis-Menten will never go higher than V max. So that is an important consideration, that any kind of data that we use needs to be in a fairly small region around the reference state. And that's what this is showing here, is that V star and X star shows that this is the reference flux and the reference metabolite concentrations, and everything else is with respect to that. Next slide, please. And so when we apply metabolic control analysis using real multi-omics measurements, and this is in a case where we did a large experiment in yeast, or actually this experiment in yeast was actually performed by the Rabinowitz lab at Yale, but we used their data. And in that case, you see this square region right here corresponds to the region where we believe the Lin log is fairly similar to the Michaelis-Menten. And of course, as you can see, its ability to predict beyond that region is quite poor, but it's certainly in the regions where we have predicted and measured fluxes, metabolomics, or proteomics, it does a very good job of reproducing the observed effects when we train the model. And the next slide, please. And so once we've trained the model using new data, then the question is, can we compute what's called a flux control coefficient? Now, a flux control coefficient is basically a ranked list of enzymes. So every single enzyme here versus a particular boundary flux. In this case, what we care about is some metabolite that we're trying to produce, but this can be applied to any and all boundary flux metabolites. So any metabolite that is excreted into the media, for example, we can calculate a flux control coefficient for. And what you're seeing here is that anything that is that is not white is basically a measurement of a flux control coefficient. And the nice thing about these flux control coefficients is that for any particular metabolite, you get a ranked list. And so there is some flux control coefficient that is highest, and that everything else is kind of with respect to that. And so you can rank the flux control coefficients and say, this enzyme is the one that will affect the boundary flux metabolite the most. And so this gives you suggestions for what enzymes you might want to modify in order to affect that boundary flux. And what's useful is that when we compare this with kind of a straw man where we just took the correlation coefficients of the boundary fluxes with all the enzymes, we see you have many more hypotheses as to what enzyme you would want to perturb in order to change the boundary flux. But in this case, we have a very sparse matrix. And so in some sense, we have much fewer hypotheses in which to test. So next slide, please. So this project was developed as part of the Agile Bio Foundry, and we have the capabilities here at PNNL at EMSL to perform this kind of analysis for any group that is interested in doing so. We would just want to work with you to make sure that the multi-omics measurements that you're making are sufficiently that there's enough samples of them that that we would give you something useful, and so we can help you with the experimental design if that's something that you're interested in. And I don't know if I can take questions or if I should just move on to the next person.
We'll go ahead and move on to the next speaker. And I just wanted to point out, we had some questions about the things that you were pointing at.
Oh, okay.
Because the pointer wasn't showing. So we will make sure that we kind of clarify some of the things maybe at the end or in a follow-up email. So just wanted to throw that out there. But go ahead.
Thank you.
Thank you.
Hello, can everyone hear me? Not sure.
Yes, go ahead.
Okay, thank you. Hello, everyone. In this part, I will talk about Peak Decoder, a new algorithm that I have developed here for multi-omics analysis of mass spectrometry data specifically. To study biological systems, there is a series of networks of molecules that interact, as Jeremy was explaining, and the tool to study these molecules at the phenotype level, it's a mass spectrometry, where we can do that with proteomics and metabolomics. And currently, machine learning is being used basically in every level of the mass spectrometry workflow to process the data, but we are working to improve that. And specifically for a alternative way of operating the mass spectrometry instrument that is called data independent acquisition to collect more comprehensive data. More specifically in the workflow to collect omics data in a mass spectrometry platform, we typically use for untargeted or global studies, we use liquid chromatography. In this case, I have proteins in the example, and the mass spectrometers operate to collect data as the samples are, molecules in the samples are separated in a chromatography column. And in the typical or standard workflow, the instrument is detecting on the fly ions, and then it's selecting the most intense ones to collect MSMS spectra. MSMS spectra is what allows then software tools downstream in the downstream analysis to assign the sequence to the molecules. And then we have this alternative mode of operating the instrument that is called data independent acquisition, where instead of selecting a specific molecules, all the molecules that elute every time point are fragmented in MSMS information is collected for all of them, which means the data is more comprehensive, but at the same time, it requires more advanced or sophisticated algorithms to be processed. The advantage of this kind of analysis is, for example, here for a study of PTMs, post-translational modifications. Since we are doing these fragmentations of all the fragments, of all the molecules, the precursors, then we can generate chromatograms, not only for the precursor, but also for the fragments. And for example, in the case where molecules coelute and they have the same parent molecule, if we want to locate specific modifications at different positions, but it's the same modification. For example, in isoforms, we can generate extract chromatograms for the specific fragments of each of the isoforms and distinguish them in the data. And this allows a relative quantitation using specific fragments for those isoforms. And more recently, DIA has also been applied for study of metabolites and has shown some improvements to detect, even to have a better coverage and better quantitation precision compared to the traditional data-dependent acquisition workflows. So now here at EMSL, we are offering in our instruments, collecting DIA type of data with LCMS, but we also have an extra dimension of separation in a platform where we have ion mobility separation between the LC and the mass spectrometry detection. And what we do, what the instrument does, while it's collecting the precursor information for each LC time point, it also adds this additional separation of the mobility. And these modes are alternated also, including a cycle where the collision energy is applied, so we can separate then the precursors and fragments based on the retention time and the mobility. This information of the ion mobility can be directly used to calculate a collision cross-section, which is a property related to the size and the shape of the molecule. And as you can see in this example, this allows to add more confidence to the type of annotations that we are doing to the molecules, separate isomers, and group also molecules, see like trends for different type of molecules. And specifically for this algorithm that is called Peak Decoder that I developed, what we are doing is taking this data and process it in multiple ways to be able to extract as much information as we can and do confident metabolite identification. And in this workflow, basically, we are processing the data first in an untargeted way, detecting, extracting fragments, precursors and fragments that coelute and have the same mobility in a process called deconvolution. And then we use those to generate also what is called decoys. And independent of the annotation of those ions, we can generate then, we can train a machine learning model to recognize, learn to recognize when the signals coelute well or not. And then once the model is trained, we can use libraries where we know we have no molecules, and then we can query the data to score this annotation and say, do I see this metabolite of this protein in the data? And then we can extract also from those peaks the integrated areas to do this comparison analysis, where we can compare different strains of different sample conditions and so on. In this example, also from the Agile Biofoundry consortium, we applied peak decoder to a study for this metabolite study. In this case, I'm showing an example in Pseudomonas putida. We quantify the metabolites, and then we compare. In this example, I'm showing we evaluated the TCA cycle, for example, in this pathway, and we compare against the results from selected reaction monitoring or targeted proteomics. In this example, the circles are the metabolites and the arrows are the proteins. And basically what we saw is that we could see a correlation, that the results agree well with what detected, what we detected in the metabolites result agree with the proteomics result well overall. So now what we are doing, it's using more deep learning. The difference between machine learning and deep learning is basically that in deep learning models, instead of telling the model what exactly are the features that it needs to use to make this discrimination for the classification, instead of doing that feature extraction by a human, basically, the model will learn that by itself. So it's processing from the raw data directly. That's the advantage. And it has been shown that using the deep learning models, in this case, the convolutional neural networks, as we add more data, the model is able to discriminate better and learn better from the data, which is not the case, and it's a limitation for machine learning models, the typical ones, the traditional ones, that they reach this plateau of during the training. And specifically, what we are building now with what we call peak decoder 2, is we are using transfer learning. We are taking a model that has been pre-trained, in this case, for object detection in images. So in this case, it's for detect a pedestrian, but then we can take that model and adapt it and tune it specifically with the mass spectrometry data training to learn to recognize this group of coeluting signals. And then what we are doing is taking the peak signals and create image representations of those to detect and score the identifications of the metabolites or the proteins. So to conclude, yes, AI/ML methods are enabling new discoveries in omics. These methods, the chromatography, the library, the algorithms, are applicable to to other omic studies, not only synthetic biology, and are available for EMSL users. The users can request these measurements with DIA or without ion mobility tool and the utilization of Decoder for their projects. And I'm now give it away to Margaret that will continue, which is our next speaker, and we will answer questions at the end. Thank you.
Thank you, Aivett. Can you see me?
Yes.
All right, great. Hi, everyone. I'm Margaret Cheung. Today, I will present how EMSL can accelerate protein structure modeling through the AlphaFold 2 software. You see, I am controlling, click, click. All right. So for the introductory purposes, first let me show by categorizing protein structures are important to systems and synthetic biology in an era of multi-omics, like what Jeremy and Aivett has been showing. So I want to convince you that proteins are workhorses of cell functions, most of you know, and the information of life is stored in a long polymer called DNA, and pieces of information passes to another polymer called proteins through the process of transcription, translation. So some proteins can fold into compact structures that can be solved by X-ray crystallography, nuclear magnetic resonance, or recently cryogenic electron microscope imaging. And in past decades, theories and computations have been developed to understand protein folding mechanisms. And the past 2 years, AlphaFold software developed by DeepMind has enabled high accuracy of structural prediction of a single protein domains with artificial intelligence and scale up the applicability to entire genomes. However, there are other categories of proteins that do not fold into distinct structures that are called intrinsically disordered proteins, and they may form fibrils and biocondensates that drive phase separations inside the cells. So for those who form structures, they can actually further build into macromolecular complexes like the ones, this is a DNA polymerase, a complex that sits on protein DNAs, and when it moves through, and another daughter strand of DNA could be synthesized like a roller coaster. So these macromolecules communicate with each other through enzymatic reactions and form biochemical networks, like those you see from Jeremy's talk. And information passes out from one node to another. It's the science of proteoforms or proteomics, like the ones you see from Aivett's talks, leading to signal transduction. And they execute cellular functions and related to many other BER's missions. So in the past few years, theories and or decades, theories, algorithms, computational tools have been developed to understand protein folding mechanism. I myself is a physicist understanding protein folding problem for decades. And a group of a research group focused specifically on structural predictions. So there's a community of protein structural prediction that happens every other year to blind test your software called CAS as critical assessment of techniques for protein structure prediction. And in year 2020, the AlphaFold 2 team from DeepMind, which is now related to Google, significantly outperformed other groups. Like you can see that there's a peak here. And from this picture, the rooming square deviation that measures the predicted structures in the real experiments on each C alpha position amino acids that really controlled within 1 angstrom, and that's very significant. So the overall template model they call TM score, comparing between the entire proteins or complex or side chain rearrangements, has been close to 1. So it means very soon that their work is published swiftly in Nature in 2 papers. One of them details their algorithms for AlphaFold softwares, and the other one demonstrates a use case saying that it can actually scale up to study every single protein that it can solve, can predict, can infer in a whole human proteome, demonstrating its accuracy and speed up. So it's really remarkable and basically sent a shockwave throughout the entire scientific communities. So why AlphaFold works so well? Well, it receives one-dimensional sequences from the right, and then it will infer or predict three-dimensional structural output with some certain confidence. And why this AlphaFold 2 significantly improved from the previous one is that it expanded its algorithms into two equally important venues. One venue is that it uses a supervised learning that basically search through the structures, learn a structure from a complete set of proteins that are being solved over the past decades. There's about 100,000 of them. And the other one that's new is to learn through evolution. They call it multiple sequence alignment, MSC, in order to correlate or learn the side chain or sequence variation and their side chains, small adjustments in spatial content. And then basically they learn through these processes in an alternative way through a very sophisticated network, and then they will predict the structures. So, and of course, they provide excellent user experience, allows everyone to use a web-based application for single domain proteins. But now in EMSL, we have it installed in our high performance computing facilities. And now you're able to not only predict one sequence, but we can receive thousands of sequences and then simulate that within a day or so, depending on how busy that a cluster will be. And this enhanced the capability for users if you receive those sequences from high throughput experiments. If you wanted to understand its structural arrangement, then you can request these additional capabilities from the proposal. So with crowdsourced testing from the AlphaFold 2 community, quickly that this web tool is taken by storm, and we're quickly aware that the limitation of AlphaFold software. And the infers accurate structure very fast, but it can't really provide the residue-specific information about their functions in a cell, which is what will be wanted. So this is where EMSL can support users to advance scientific discoveries by providing structural models for developing hypotheses and research designs. So I want to show my last slides because it echoes the first one. It showcases the challenges in research design for connecting molecular functions to a cell's phenotype, because the complexity as a y-axis grows from genome, DNA, RNA, proteins, and interact them to phenotypes. So even though AlphaFold is great tools to predict the structures, but it didn't tell us what they do to within one another and how they were modified. So with EMSL, we have leveraged streamlined user experience through open on demand. You don't have to be an expert in high performance computing. You can use it that as a part of the workflow in your data analysis, either that's from mass spec or from NMR study. And we tie that back end that you don't have to see, but the job is actually executed in high performance computing Tahoma. So in your proposal, you're going to request how much hours you wanted. If you don't know, that's why you can ask Jay or ask Satish, our P leader. We leverage experimental data that most users upload to Nexus and streamline workflow from structural characterization, imaging, high throughput to structural modeling. And of course, within the structural models I mentioned, not all proteins have structures. You may require additional help from computational scientists like myself in order to help you understand and interpret what those variation means. So in your proposal, you can request staff time as part of the resources. Okay, and any questions? If not, I'd be happy to address. And I think Megan has posted my contact information on the chat, and I'd be happy to take more Q&A questions at the sessions. Thank you so much. And my Google Scholars profile is linked. You can see the type of my research relating to protein structure modeling, molecular dynamic simulations, quantum modification of side chains, to all the way to protein interactions in the cells and imaging. I can do a lot. Thank you.
Great,
You can
thanks,
take
Margaret.
the control.
Yeah, I was gonna say, you can keep your camera on. I'll have the other presenters join us so we can launch into our Q&A portion. And Margaret, I think we'll start with you. Since we did have a question come up from Michael Green.
Michael.
His question is, can AlphaFold predict a change in conformation with application of an electric field?
No. Anything that deal with dynamics, you need molecular dynamics. So AlphaFold is to infer a gas structure. It provides some static information, picture, really nice, as a starting point for you to think about what it could possibly do. And this is a part that EMSL scientists like myself can help you.
Great.
Or advance your research.
Great. Okay, question for Jeremy from John Cliff. He's referencing during your presentation, are these models strictly mathematical, or are they constrained by potential mechanistic constraints? For example, in your example of a product slowing rates, is the type of inhibition explicitly modeled, or are you fitting a curve that models the data well, regardless of mechanism? Hope that makes sense.
Yes, like, hi, John. This is Jeremy. Yes, our priors are based on mechanistic knowledge of which metabolites or reactants or products of which reactions. And the way that we do that is, so that mechanistic knowledge is derived from a metabolic reconstruction of an organism, such as a genome scale metabolic model. And the way that it works is that we use deterministic equations that are derived from the metabolic model, but there are parameters that we don't know about. And so the uncertainty in those parameters is represented by those priors. And we sample from those priors, and we run the, and then once we have priors for all the, we have a sample for all the parameters, we run the equations and check to see what this steady state solution is. And we compare the steady state solution with any multi-omics measurements that we've made, because we can predict what the proteins look like, what the metabolites would look like. And if it's not a good match, then it means that we need to reject that sample and try again. And rather than doing this kind of rejection sampling that I described, we use something called automatic differentiation variational inference, which is a gradient-based method to find the posterior distribution. So very efficiently, it can find a set of parameters that can reproduce the multi-omics measurements that we have measured. And then it can go one step further and calculate these flux control coefficients, which is basically a sensitivity analysis on the on those parameters that tells you, hey, if I increase the enzyme this much, and that will increase the flux of my bioproduct by this much. And so in this way, we can suggest improvements to make to your system.
Thanks, Jeremy. Ann Summers actually has a question for you. Her question is, how are side reactions handled?
So I'm not sure what you mean by side reaction. Do you mean like cofactors of a reaction?
I am going to allow her to talk so that she can ask you. So hold on 2 seconds. Okay, Ann, go ahead.
Thank you. Can you hear me?
Yes.
Okay, thanks. I was thinking about the, I mean, you're looking at a particular metabolic pathway of interest, but the cell's going to be doing whatever it wants to do. And it is the case in all of biology that you take a whack at a system and suddenly you find yourself getting kicked in the behind by something else it's doing.
Yes.
So I just wondered about, how you guys handle that.
No, that's a really good question. I think at the very beginning, I said, we only, if you try to do any like large perturbations to the organism, like you knock out a gene or you add a brand new enzyme, then it's basically going to move the system out of something that we can compare against. So we have to have some highest performing strain, and then everything else is like small modifications of that. And then what we do is we just give you like a nudge in the right direction. So if we say if you increase, so the idea is that this is a way of performing incremental changes to the system. If you need to perform more large scale changes to the system, like you've got a tighter, this is the case where you have like a tighter rate, you have a titer of I don't know, 30 grams per liter, and you need to get to 35 grams per liter. It's just like, how can we nudge it in the right direction? If we try to,
well, yeah, but how do you look for those things that are unanticipated and could be, even with a very, my experience with biological systems is that you can take something up 10% and get really weird surprises. So anyway, I don't want to keep going, but I
think
I wasn't asking how hard you were hitting it or how hard you were tweaking it, but how, is somebody watching for things, or is your algorithm watching for things that, woohoo, look what we just did?
Yeah, well, so there's a couple ways in which we do try to take into account things that we did not anticipate. So we have this prior knowledge of the metabolic network, but we also allow for the possibility that there may be additional gene regulation that's going on. And so, or additional allosteric regulation that's going on. We're not making any claims as to what the mechanistic source of that regulation is, but we can certainly detect when there's weird kinds of regulation that we wouldn't have expected.
They might be good. Yeah.
Yeah, exactly. Yeah.
Okay, question for Margaret from Young Kin Zhao. Hi, Margaret. Do we need to submit an EMSL proposal in order to use the on-demand protein folding platform you have at EMSL without necessarily involving EMSL staff help?
That is correct. If you wanted to use any resources, you should submit a proposal, but you don't need my help. The things that are being installed on Tahoma is quite straightforward. So you've had extensive experience, then you do not have to request the staff help. But if you need staff help, then we are here. Thank you. And Jay is here, can further elaborate my explanation.
Nope, you nailed it, Margaret.
Thanks, Jay.
Okay, another question from Anne. This one is for Aivett. How do AI conclusions compare to what is known from conventional methods? For example, what are the positive and negative control systems?
Yeah, thank you for the question. So far, we have compared at the level of the number of molecules that we can detect. We compare against another software that is called MSDial, and we could identify more than double of the metabolites that MSDial could identify. I didn't show those results here. And we compare the quantitation performance to other methods, to using selected reaction monitoring. And we, using this platform, in this case with the ion mobility, we can detect even a lower concentrations, one more order of magnitude. And we compare also the annotations against a manually curated data set. And for about 200 annotation of big groups, we missed only 4. So the performance is really much better compared to the conventional workflows.
Great, thanks, Aivett. Jay, question for you. Do I need to know exactly what I want help with? Suppose I have a biology problem I'm trying to understand better, and I think simulations or modeling or data science could help. Is it okay to reach out?
Yeah, absolutely. We are in a period of rapid growth. For a long time, computing specifically supported the 2 other science areas and was embedded in them. But with the proliferation of high performance computing and the entire community of communities, plural, of computational scientists, modelers, data scientists, AI, ML, all of that, there's just a lot of opportunities for cross-fertilization. If you're developing data analytics techniques and want to try them out for identifying outliers in EMSL data, great, give us a call or send us an email. If you are studying a particular system and you think that certain kinds of modeling might help, but you don't even really know what that modeling might be, please email us. Margaret would love to chat with you. I would, we have others. And likewise with metabolic modeling or analysis and understanding of mass spec data. Pretty much if it's in the scope of biology and environmental research outside of human health interests, and it involves computing, and you're not entirely sure, send us a line and let's talk about it. We'd love to hear what you're excited about and if there are ways where we might be able to advance your science.
Great. Thanks, Jay. That was a good explanation. Margaret, how confident are these inferred structures?
So the structures for AlphaFold, they actually assigned confidence according to their algorithm and data. So if your structure has existed in the protein database, the confidence is nearly 1. They call it TM score. It's called template model. But if the protein does not have a structure, like intrinsically disordered proteins, or the structures has allostry, then it's probably you're never going to see any like a folded component in their map. Then confidence is nearly 0. It's not a magic bullet, so you still require scientists to hash out residues to specific information.
Can I ask a question?
Oh, go ahead. Sure.
Okay, yeah, thank you. I had posted it, but it fits so well with the one that you just answered. I was wondering to take it to another dimension. I mean, in AlphaFold, crystal structures and to some degree, MRI structure, I mean, dynamic structures that are done in solution are considered, but neither of those things actually may work inside the cell. And I'm wondering to what degree anybody is considering, or maybe the algorithms already do consider, genetic mutations that people are aware of, or other things that would influence the real biology, which is actually only ground truth with mutants. So that was my extension of the question earlier.
Excellent question. And my research domain is actually protein structures in the native environment. So I completely agree with you. And if you're interested in sending a proposal on trying to interrogate some specific residues, I think there's lots of opportunities here. And in the past, I think one of the funded EMSL proposal is looking at that. So definitely it's an area of interest that we can leverage a lot of high throughput capabilities at EMSL. We can do multiple sequences and at the genome level and trying to perturb that information and then feed to the structure. And integrating with molecular dynamics and many of these fancy tools we have developed over the decades, I think we can help the users advance scientific discovery.
Oh, glad to know that.
Can I expand on that, Margaret? There's something that I think there's sort of a coherent set of interests. And per your question, clearly a lot of proteins are post-translationally modified, and identifying those and modeling those is something that Margaret and others at EMSL and around PNNL have a lot of expertise and interest in. And identifying those post-translational modifications is something that we have a lot of experimental expertise and data analysis expertise with as well. So there is there is an integrated sort of capability in that space if you're interested.
Wow, thanks.
Yes, so we're going to try to put a pipeline. So starting with AlphaFold structures and then decorate that with a lot of experimentally interested modifications and see how that impacts the property.
I was actually thinking of the informatics actually that's already existing out there.
I'll be happy to know more.
Okay, sure.
Great. That was a good conversation. Well, I just want to encourage anyone who didn't post a question to reach out to any of the speakers here or any of the integrated research platform leaders with any questions that you might have going into this next open proposal call season. Be on the lookout on social media and on our website for calls that will be opening very soon. And with that, we want to thank you for your time today and hope to see you at our next EMSL Learn webinar. Have a good day.
Thank you.
Thank you all, and thank you to our speakers today. Take care.